So in today's class, I will be talking about amino acid and proteins. So people must have definitely heard about amino acids, and you know that they are one of the essential biomolecules which make up our body. That is more significant because these amino acids, they join together by a type of bond known as peptide bonds to form the proteins. And proteins are the most important components of our body. They are essentially the enzymes. They actually they perform several all the activities inside our bodies, all the reactions in our body. They are produced by enzymes. Not only that, all our skin, flesh, everything, most of it, it's majorly proteins, proteins everywhere. So if there was no proteins, possibly there would have been no life. So the most important component of life, in my opinion, personal opinion, is proteins. People call DNA, RNA, nucleic acids as important. They are very important. They definitely. And theory is that possibly there was a battle. If you talk about the origin of life, there was a battle whether which came first, the proteins or the nucleic acids, which came first. And so the debate continued, and right now things are tilting towards nucleic acids. They came first because they found a nucleic acid which had enzyme activity, the ribozyme. So after that, this proteins actually they lost out the race of being coming in first. Many people thought that proteins were nuclear were the genetic materials, but eventually that idea got discarded. So when this idea got discarded, uh, people then it got discarded because they actually it was proven by mm, Griffith and Avery McCarthy, MacLeod, and all these transformation experiments and things in the classical experiments in genetics. They proved that the DNA was the genetic material, and then RNA was found out. So everybody wanted to believe that mm, DNA was actually the uh, origin of DNA was actually the origin of life. Actually, the DNA material, it came first, and because of this DNA, life came. Everybody was of this concept, and eventually, the first molecule was RNA. It was found out to be it was assumed to be RNA because RNA with an enzymatic activity which could perform all the reactions, it was found. So RNA then DNA, they were the nucleic acids. They were losing the war of being the first biomolecule, was because of the simple fact that it did not have a enzymatic activity. But the moment enzymatic activity was found in RNA, everything tilted in favor of nucleic acid. Okay, they are the first materials to be formed. But amino acids, they all aggregated. We will talk about origin of life later when you start talk about evolution. But today we will therefore study a very fundamental basic biomolecule, that is amino acids, and the joining of amino acids together to form proteins. So with this, let me I'll share my screen. উপরে তুলে দিই। And now where is the screen? Please do tell me if this is visible or not.
Yes, sir.
It is visible. Great. Okay, let's talk about the first amino acids. So what are amino acids? Basically, they are the building blocks of proteins. So right from bacteria to higher organisms, all organisms, they have 20 standard amino acids. So there are 20 standard amino acids. Apart from that, there are other amino acids, but these 20 standard amino acids found in all organisms. They are found in all organisms. So out of this, 19 of them, they are alpha amino acid. I will explain very shortly what is alpha amino acid. And one amino acid, its name is proline. It is an amino acid. It has got a structure so difficult. I mean, it's not difficult, I would say, but it is my inability to remember the structure even after teaching for so many days. There are three amino acids which I cannot remember the structures. One is histidine, tryptophan, and the proline. So these three amino acids, you have to be very careful of remembering the structure. Three amino acids whose structures are very difficult to remember. For me to remember. The first one is proline. The second one is histidine. And the third one is tryptophan. Even I also get nervous if they ask me in an exam to write these structures. I get nervous. So you people try to remember these structures properly. So now we are talking about the rest are simple. What is an alpha amino acid? Let's draw the structure of an amino acid. It has got an N amino group. You can basically draw it as NH2. There is a carbon. Then you have a COOH, H, and there is an R group. This R group can be, this is the side chain group. So I'll draw the structure once more in a different manner. Amino acids normally in a medium where there is a lot of hydrogen ions in an acidic environment. Basically, lot of H plus will become saturated with H plus ions, and less of H plus, that is more alkaline environment, will have less H plus is attached. So this amino acid, a normal when charges are balanced, I will explain this right now. Amino acids exist in a form called Zwitterion. So this form which I draw over here, it is a Zwitterion. It can be one extra hydrogen can stay here. It has got a positive charge. So the net charge is +1. In an acidic environment, H plus is lot. So all the H plus goes and sits over here. In an environment for this particular amino acid, for a particular amino acid, when this becomes there is net charge is 0. This is a Zwitterion. And in an acidic environment, alkaline environment, it can very well be all the H pluses are stripped away. You have a negative charge over here. The charge is -1. So this happens in an acidic environment. This happens in an alkaline environment. So here pH is alkaline, and the pH at which net charge is 0, that is known as Pi, isoelectric point, for this amino acid. Is it clear so far?
Yes, sir.
Okay. Anybody needs an explanation again? Yes, no?
No, sir.
Okay. Now we will go. Let us see the amino acid. I'm going to draw it in a very peculiar form. H3N, I'm drawing. You can write NH2 over here. It's how you have practiced. I have practiced like this, so I'm drawing it like this. I'm drawing a CO minus. Charges are balanced. Plus is balanced, minus is the charge is balanced. This is the side chain R group. This carbon which is attached to four different groups, is known as the alpha carbon. Now, in the case of carbohydrates, you had seen if the OH was on the right side, it was a D carbohydrate, and if the OH was on the left side, then it was an L carbohydrate. In the case of amino acid, instead of OH, we talk about this H3N plus. So if this is on the, if on left, it is an L amino acid. If on right, it is a D amino acid. So basically, if you draw something like this, this is a mirror image of each other. So this is on the left side, so this is an L amino acid. And this is a D amino acid. Am I clear?
Yes, sir.
Akina?
Yes,
Okay.
sir.
Okay. So this is an L and D. And these are mirror images of each other. In carbohydrates, you had seen that the D carbohydrates more abundant in nature, and in living system. But L amino acids are more common in living systems. In fact, all of them, there are one or two exceptions where you find D amino acid in living systems, but mostly they are all L amino acids. Now, one thing which I will explain are the different types of amino acids which are present. So 20 is standard types of amino acids are present. So 20 standard amino acids can be divided into first one. It is the hydrophobic, aliphatic amino acids, aliphatic means straight chains. So let's draw some aliphatic amino acids, hydrophobic. Hydrophobic means it hates water because of a hydrophobic chain. So this structure remains the same. H3N plus, draw the alpha carbon, COO minus, 1H. Only the R group changes. If the R group is a hydro H, then this is a molecule of glycine. Now, one more thing which I forgot to mention over here is that this C over here, it is attached to four different groups, and therefore this carbon is a chiral carbon. Are you people clear with this point? Yes, no?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
So this becomes a glycine. Now, if instead of in glycine, it is not attached to four different groups. So this carbon is not chiral because you have one hydrogen attached over here and second hydrogen attached over here. So it is attached to three different groups. So this is not a chiral carbon. Now immediately, if you, for example, if you go to this remains the same, and immediately if you attach, instead of this hydrogen, you attach a CH3 group. This R group is a CH3. This becomes an alanine. Again, if you draw it, you try to make groups, you practice these structures 3 times, you will be able to remember it throughout your life. At least I can do it. If you attach a CH over here, if you attach this R group over here, this group, this structure becomes a valine amino acid. Like this, you can draw several of this leucine. Proline is difficult. I am unable to remember proline. The next type of amino acid which we will be talking about is the hydrophobic aromatic amino acids. Draw it the same manner, no problem. You draw it. If you draw it like this, in this R group, simply attach a phenol group like this. This becomes a phenylaniline. Again, if you attach here a phenol group, this becomes a tyrosine. This amino acid, this OH, it's a target for phosphorylation. This OH. Tryptophan is very tough. I'll forget. I don't remember it. The next type of amino acid which we will talk about are the polar charged amino acids. So these actually are hydrophilic in water. The others were hydrophobic, soluble in water, and are polar. That means positive charge is concentration high in one end and negative in one end. Something like this. This is known as polar. This is a polar molecule. So let's draw the structure of it can be positive and negative. Let's draw the top positively charged ones. Positively charged. Who are the positive charged ones? The famous ones are the arginine, lysine. Let's draw them. That's not very difficult. Arginine has got 1, 2, 3. Okay. একটা দুটো তিনটে কার্বন থাকে, তারপরে নাইট্রোজেন থাকে, তারপরে একটা NH2 থাকে। কার্বন থাকলেই always there is an H2 is always attached. N has got 3 arms. There's supposed to be 1 carbon more. এখানে NH2 plus. N has got 3 arms. 1, 2 are carbon over here. And since that extra hydrogen is arm, the plus is there. This has got 4 arms. There's supposed to be an NH2 group over here. Now, why is it positively charged? +1, +1, -1. So net charge +1. Lysine is the same. It doesn't have actually this group over here. You have instead of this পরপর 4টে CH2. Alpha carbon COO minus H 1, 3, 4 and followed by NH3 if it is a plus, and whenever there is a C you add H2s over here. Practice this 3 times looking and the fourth 1 time without looking. You will remember it throughout your life. I've studied it 20 years back. I still remember it. Although I'm teaching now this for the last 7 years regularly, that's made it more easier. But this was not tough to remember. Here's a plus charge. Here's a plus charge, negative. So it has got a net charge plus 1. Is the structure clear? Yes, no?
Yes, sir.
Okay. So these were the positively charged. Let us now talk about the negatively charged amino acid. So among the famous ones are the especially the most famous is the serine, threonine. So they are also targets of phosphorylations. You will later realize that several enzymes, in order to be active and inactive, they are phosphorylated or de-phosphorylated for their activity uh, in catalysis. You draw it similarly, COO minus. You just draw a CH2OH, an alcohol group over here. This is serine. And in threonine, it becomes a CH, CH2OS, CH, or CHOH, CH3. It's just a bit different. I'm always forgetting the plus charge. You must always like the plus. Nitrogen has got 3 arms, but if you are showing a fourth arm over here, then there must be an extra hydrogen. So it becomes NH3. You have to give NH3 plus. It takes an extra positive charge. Here it becomes COH, CHOH, CH3. So this actually to be fairly speaking, this OH over here and here, they are the targets of phosphorylation. So all these amino acids, they are polar, these are uncharged amino acids. Next, we will be dealing with, which we should have dealt before. Suddenly, I wanted to show you the targets of amino acid phosphorylation. We will go now to negatively charged. So, who are the most negatively charged? Can anybody of you say who are negatively charged amino acids from your general knowledge? Just tell me whatever you know about it. বলতে পারো? নেগেটিভলি চার্জ অ্যামাইনো অ্যাসিড বলতে পারো? জানো?
Sir,
Yes, no?
glutamic acid.
Exactly. Aspartate and glutamate. Very good. কে বললে এটা? Pernally?
Yes, sir.
Okay, very good, very good. এটাকে আমি পুঁছি খুব বিচ্ছিরি করে ফেললাম। I made it very dirty. So, most famous are the aspartate and glutamate. It is very simple to remember. CH2, just add one CO minus over here. So you have a minus charge here and a minus charge here and one plus charge. So minus one, net charge minus one. So this is aspartate. Glutamate has got just one more CH2 here. This is glutamate. Clear? Are you clear about this? These negatively charged amino acid,
Yes, sir.
and they are polar. You have one plus side over here and one negative side over here. Therefore, they are polar. So now we will go to an important part, that is peptide bonds. Let's get back to the white color. So peptide bonds, proteins, polypeptides, chain of polypeptide. It is made by linking several amino acids together with the help of peptide bonds. So polypeptides together, many polypeptides together, many of them may form a protein. Many of them or the same of them. So let me draw for simplicity. I'm drawing the amino acid structure a bit differently. I'm drawing the hydrogen on the top. This is the first R1 group over here. CO O minus. Instead of the COOH, I'm drawing it like this. This plus another I'm drawing. This is R2. Two amino acids together. Can you understand this? Yes, no?
Yes, sir.
Yes, sir.
So they are going to get attached now. R1, double bond O, this region is hitting. I'm drawing this region. CO, I'm drawing it on the top. NH, CH, R2. So this particular CONH region, this is a peptide bond. Clear? Plus a molecule of water is released. Clear about this?
Yes, sir.
Yes, sir.
Now, if we look at the nature of a peptide bond, you will see I'm drawing this CONH. This, there is an electron. Sometimes it migrates over here. This type of a phenomenon is known as resonance. You can find it in a benzene molecule. These electron pairs, they keep on migrating from here to here to here to here. They keep on resonating, moving. So therefore, it is better to draw benzene like this in order to show a resonance. The structure of benzene is where the electron pair keeps on migrating. The structure of benzene was elucidated by Kekulé. This phenomenon of resonance, he gave this phenomenon. He dreamt that two snakes, one snake was like this. It was eating another snake. So one snake was keeping on eating another snake. So he dreamt it like this, and then he came to across the phenomenon that these electron pairs, because suddenly benzene's reaction was showing as if the bond was present once over here, once over there. If the polarity, the optical structure of benzene, it was showing something like this. So he was baffled by it. So one night he dreamt one snake was eating another snake, and that snake was being eaten by another snake. So one was eating another, that was being eaten by the first snake. So from that he came to the concept of resonance. So from there you have that famous thing which he said, "Let us learn to dream, gentlemen." Because and then work to make this dream into a reality, and it's very important. We must in life we must learn to dream. I get excited when I hear these how these discoveries are made. If you people are interested in the history of science, these are very fascinating things. Life of Mendel, life of these famous scientists, you could read through them. They were exceptional men. They were normal men who became exceptional, I would rather say, from simple facts which they see every day, but then suddenly it becomes so obvious to them, they make a rule which makes the changes the world. Like Mendel, a simple statistical calculation that actually led into a particular brilliant genetics. Or you would say Thomas Hunt Morgan, not brilliant, all simple experiments which make huge results, discoveries. So sometimes this electron pair, it jumps from here to here. And the fascinating part of this peptide bond is then this becomes something like this. 。 . 。 。 . 。 . 。 . 。 . . 。 . . 。 . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . 。 . 。 The double bond has migrated over here, and then it can sometimes go back over here. It can sometimes come over here. This becomes a double bond. So you can see this is almost like this single peptide bond which you are seeing over here, CONH. This this has got a partial double bond nature because of the resonance of this particular electron pair jumping from here to here. It has got a beautiful jumping nature, and it's got a part. It is, you can say, this peptide bond is stronger than a single bond. It is weaker than a double bond. So people call it a partial double bond nature. The next part is a bit, maybe a bit difficult for you to visualize, but anyway, let's draw the structure. It has got a rigid, planar. It's rigid and planar in a single plane. Planar means it lies so if I draw it something like this, let's draw it slow, just in a bond which I'm showing in H. CH R1, there should be the CO over here. NH, this is the peptide bond I'm drawing. I'm going to the next carbon. NH, এটাকে উপরেতে CH R2, এখানে CO. This is you are going so on, so we are concentrating on this thing. Let's I cannot draw it. Let's draw the third one over here. Let's draw a third R3 group also. CO NH, এটাকে একটু এদিকে সরিয়ে নিই। CONH, তারপরে CH R3, now I can draw it. Now I use a different color. So I've drawn these boxes like this. What could be the reason I've drawn these boxes? They are rigid and planar. A single plane. They are in a single plane. Single plane. If I draw this line over here, you cannot twist and turn them. The whole thing, if you have to twist and turn them, you have to, it will move as a plane like this. So the, if it is like this, if you twist and turn them, it will be plane. So it will move in a plane. You draw it like a CO. Huh? This is auto correction. So it moves like the plane. If this is a single plane, CO, why is it drawing these boxes? Anyway, whatever, whatever is happening, I don't understand this. And this is a whiteboard problem. I don't want CO in H. If I draw it something like this, and then draw a plane. So the CONH, if it rotates, it will rotate in a single plane. So if you draw a single line, it has gone with an angle. It has rotated into an angle. There is an angle of rotation, and this rotation will be at in a single plane. Is it clear? বুঝতে পেরেছো?
Yes, sir.
Okay. So this is one part of it. And now let's draw what are these angles. This is C alpha. The alpha carbon, I'm not, for simplicity, I'm not drawing, for simplicity, I'm not drawing the H and all over here. This is the next carbon over here. This is, I'm not drawing the double bond. This is an oxygen O over here. I am drawing the nitrogen CON H. This is the peptide bond which I am drawing. And this is attached to the next C alpha, the central C alpha carbon. This is attached to the R group, and it will be attached to a hydrogen. This is attached to the next C, O. C alpha again, H. So this bond, this angle, this is one plane. This is another plane. Let me do it with another color over here. I'm trying to explain the plane. This is another plane. So this angle which is rotating, this is the between the N and C. This is this particular angle. And this angle which is rotating, this is called this phi bond. One is psi and one is phi. So the C alpha N and this is the this bond, and this bond C alpha C, this is the psi bond. So this is an angle. So basically, since it's a plane, one plane is here, one peptide. This is my whiteboard problem again. This is one peptide bond. It's in one plane. Another peptide bond in another plane. And you can see, if you draw a straight line through it, there is an angle. One is this, and the other is this. Clear so far?
Yes, sir.
So basically, can you see my hands? You people, can you see my hand?
Yes, sir.
So one is here. हाथ दिख रहा है एक सर, एक तरफ से हाथ हाथ इधर घुमा दिया. Okay, let me show it with two mobile phones. This is one angle. This is another angle. One peptide bond is like this, and one peptide bond is like this. दोनों के बीच में, can you see an angle between both of them? ये तो कोई एंगल नहीं है, लेकिन जब घूम जाता है एक एंगल दिख रहा है ना बीच में. Yes, no?
Yes,
Okay. So it's something like this which is happening. So the angle between the C alpha and an N, that is the this particular symbol over here. ये वाला एक है, ये वाला और एक है. This is another one. So and this actually depends, this angle, on the R group which is present. ये clear है? Is this clear? And this is because, say this is then one amino acid going, or a polypeptide chain going. This is R1. This is the next R group coming up. This is the R3. R4, here you have the NH3 plus. Maybe here you have the CO minus. It's going like this. So this can be hydrophobic. These R groups. they can be hydrophobic, they can be hydrophobic aromatic, aliphatic, they can be hydrophilic charged. So these R groups push and pull among each other. Push and pull, that is, they attract or repel. If you take two magnets, for example, two magnets, one side north pole north, if the north and north face, they will push each other away. North and south face, they will pull each other away. That's how a magnet works. Similarly, if two positive charges, they will repel each other, and a positive and a negative charge, they will attract each other. So like this, they will push or pull each other. They can push and pull each other. Result is these angles are formed, and based on their push and pull, all the proteins will take a particular structure. This type of a structure is known as alpha helix. If the protein structure, because of the R group, makes it coil like this, here at the end there are all R1, R2, R3, all they are coming and coiling with each other and going. This is known as alpha helix. If it goes like this, say this is the direction N group, if it goes like this is known as beta sheet, and this region where it turns, it is known as beta bend. So basically two types of structures of the most important structure. One is the alpha helix, the other is beta sheet. Alpha structure and beta structure. These type of basic structures of the proteins are formed. Clear?
Yes, sir.
Okay. So now we will go discuss the last part of this, that is what primary, secondary, tertiary structure is. So primary structure of a protein. So the primary structure of in a protein, it is the linear sequence. It is the linear sequence of amino acids as joined by peptide bond. And this is dependent on sequence of nucleotides. On DNA. So DNA You have a DNA molecule. This will form a molecule through a process known as transcription. I have sent the overview of gene expression note yesterday, small, through process of transcription. It will form RNA or something specially known as messenger RNA. And in message, here you have nucleotides. AUG, blah, 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 blah. It will go like this. So AUG, if, basically it is ATG, basically. In mRNA, it will be become the other way around. And so this will code for the amino acid methionine. So a triplet of nucleotide rather I would say a specific triplet of nucleotide will bring a specific amino acid. So depending upon this code, nucleic acid code, which is present on the RNA and eventually on the DNA, you have a sequence of amino acid. Is this clear?
Yes, sir.
Okay. Now, several amino acids like methionine, NINE, cysteine, they have got sulfur groups, SH groups. So this is an amino acid which is this is a, let's cancel this. So this is a protein polypeptide and it has got an SH group over here. This has also got an SH group over here. So they can form a structure. They can get oxidized and form a structure like this. We have read this in pentose phosphate pathway. But under primary structure, say this is a particular protein. It has got an SH group over here and it has got another SH group over here. Now, what can happen is this protein can fold so under primary structure, intramolecular within the same molecule, this is known as a disulfide bond. Within the same molecule, in this case polypeptide, plus linear sequence of amino acids. They all fall under primary structure. Clear? Yes, no?
Yes, sir.
Yes, sir.
Okay, thanks for you are getting to understand. Now, what I have explained, the under secondary structure, the, today this pen is giving a lot of trouble today. It seems I have to buy a new one. So, because of these R groups, you have the C alpha. This is the C alpha here, C alpha another. Here is a C alpha. So these are the consecutive amino acids. So this particular here the distance is between this fold, it is 0.15 nanometer. This side is 0.54 nanometer. The second alpha helix and beta bend, beta sheet, they because of the R group, these are formed. Beta sheet depend on the R group of amino acids, how they repel and organize with each other. And so this is the secondary structure. Now we will go to the tertiary structure. One polypeptide this is another polypeptide, say in hemoglobin. This is alpha chain of hemoglobin, this is beta chain of hemoglobin. So it can happen that hemoglobin, so it can happen that they can be associated like this. There can be a hydrogen bond, disulfide bond, two different polypeptides can associate and form a protein. This is known as the tertiary structure. Most famous example is that of hemoglobin. Let's use some orange color, a BJP color. We have been using Trinamool color a lot. Let's use a BJP color now. So it is made up of two alpha subunits and two beta subunits. These are say two alpha subunits, two beta subunits. They are associated together through this mechanism. Such thing is known as quaternary. So, this type of structure when present, this is the tertiary structure. When two different polypeptides come and associate, these are all part of quaternary structure. And in a single molecule, it takes a particular native conformation. A single polypeptide takes its functional native conformation. This is the tertiary, 3 number structure, tertiary structure. So if I can draw it clearly, it can be, this is an alpha helix part. Then suddenly there can be a beta bent part. Then again a region where there is a proline, there is a helix breaker. Again an alpha helix part can come. Again, maybe there is a beta bent part. It can very well be that between this part and this part, I draw with another color, there is some hydrogen bond, disulfide bond, or any other associative bonds. Any other association. So this type of a structure is the tertiary structure. Clear? Tertiary structure কাকে বলে?
Yes, sir.
Okay. And quaternary structure, to differentiate in quaternary structure, so alpha beta are there, so there can be associations among them. To get these associations are there, and to get native structure. Clear? And in quaternary structure, two different polypeptides are involved. Two or more. Like in the case of hemoglobin. One chain, it associates with another chain, say for example. So all the associations between the two polypeptides, the two কি হলো? হ্যাঁ, টু different polypeptides. Okay, so this is the end of this protein chapter. So I will stop my sharing today. And any questions?
No,
No,
sir.
sir.
No questions today. Tired? কালকেও একটু লম্বা হতে পারে যদি কালকে দিতে দেয় আমাকে কলেজ। ঠিক আছে, তারপরের দিন একটু কমে যাবে আবার আস্তে আস্তে। এই দুটো লম্বা চ্যাপ্টার আমি শেষ করতে চাই তোমাদের পরীক্ষার আগে। ওকে দেন, আই এম স্টপিং দ্য রেকর্ডিং। করবো তো?
Okay, sir.
ওকে।